Hello, friend. Welcome to Science for Sleep. Tonight, we're going to spend some quiet time with something small, something bright, something that often fits in the palm of your hand. Berries. They grow low to the ground or hang lightly from thin branches. They appear in clusters, often hidden among leaves. A sudden patch of red or deep purple, or almost black-blue against green. For most of human history, noticing that color shift in a landscape meant something important. It meant food. But before we wander too far into forests and fields, there is a small surprise waiting at the very beginning. The fruit many people think of first when they hear the word berry is the strawberry. Soft, red, dotted with tiny pale seeds. And yet, botanically speaking, a strawberry is not actually a berry at all. In plant science, a true berry develops from a single flower with one ovary, and the entire fruit is formed from that one structure. Blueberries are true berries. Grapes are true berries. Even bananas qualify. But strawberries are something else. Each of those tiny specks on the outside, the little seed-like dots, is actually its own small fruit, called an achene. The fleshy red part we eat is a swollen flower base holding dozens of these tiny fruits together. It's a gentle reminder that the words we use in daily life don't always match the quiet rules of biology. And if you enjoy these quiet journeys, you're welcome to subscribe. And if it feels natural, to leave a comment sharing where you're listening from tonight. It helps others find their way to this calm corner of the world, one sleepy traveler at a time. The quiet continues here, just as it is. Strawberries may not be true berries in the botanical sense, but blueberries are. Small, round, deep in color. If you roll one slowly between your fingers, you might notice a faint, dusty coating on the surface. That soft bloom is part of what makes them appear blue. Underneath, the skin is darker, closer to purple. Blueberries have drawn attention not only for their color, but for what researchers have been studying about them. In recent years, controlled trials have looked at how regular blueberry consumption may affect certain aspects of memory and processing speed, especially in older adults. Some studies suggest improvements in specific cognitive tasks after weeks or months of supplementation with blueberry powder or extracts. Scientists believe compounds called flavonoids, natural plant chemicals that also give berries their color, may support blood flow in the brain. The evidence is still being explored. Research continues, but the fact that such small fruits are being studied in connection with memory and attention is quietly interesting. For early humans, berries were not research subjects. They were seasonal signals. A flash of color in summer meant quick energy in the form of natural sugars. It meant vitamin C, which the human body cannot produce on its own. It meant hydration wrapped in thin skin. Compared to meat, berries are light in calories. Compared to roots and tubers, they are easier to gather, but only when they are in season. Their sweetness is not an accident. Plants evolved fruit to be eaten. In exchange for sugar, animals carry seeds away from the parent plant, spreading the next generation. So even at the beginning of our story, we see several quiet layers. A fruit that is not technically what we call it. A pigment that may help plants manage stress and possibly help support human health. A survival trade between plant and animal that has been unfolding for millions of years, and all of it contained in something small enough to rest on your tongue. The difference between what we call a berry and what a botanist calls a berry becomes clearer when we slow down and look at how fruit forms in the first place. Picture a simple flower in early summer. At its center is a small structure called the ovary. After pollination, when pollen reaches the flower's inner parts and fertilization occurs, that ovary begins to change. It swells. The walls thicken. The tissue softens. What was once a flower gradually becomes a fruit. In the case of a true berry, that fruit develops from a single ovary in a single flower. The entire structure, skin, flesh, and seeds, comes from that one starting point. Blueberries follow this pattern. So do cranberries. Grapes do as well. Even bananas, which many people never think of as berries, fit the botanical definition because they form from one flower with one ovary that ripens into a fleshy fruit containing multiple seeds. Though in cultivated bananas, those seeds are now tiny and underdeveloped. The shape of a true berry is often round or oval. The outer layer, called the exocarp, becomes the skin. The middle layer, the mesocarp, forms much of the soft flesh. The inner layer, the endocarp, surrounds the seeds. These layers are not visible in a dramatic way when you bite into a blueberry, but they are there, arranged in thin, organized sheets of plant tissue. Now return to the strawberry. Its surface is dotted with those small, pale specks. Each one is an achene, a tiny, dry fruit with a single seed inside. Instead of forming from one ovary, the strawberry develops from many ovaries within one flower. The red portion that swells and sweetens is not the ovary at all. It is the receptacle, the part of the flower that holds everything together. It enlarges and becomes soft, carrying dozens of individual fruits on its surface. Raspberries and blackberries follow yet another pattern. They are known as aggregate fruits. Each little round bead in a raspberry cluster is called a drupelet. Each drupelet develops from its own ovary, and together they form the larger fruit structure. When you pull a raspberry apart gently, you can see those separate units clustered around a hollow center. These differences are quiet and structural. They do not change the taste very much, but they reveal how plants organize reproduction with careful precision. The number of ovaries in a flower, the way tissues expand, the way seeds are positioned. All of these details shape what we eventually hold in our hands. This organization also explains why some fruits that seem nothing like berries are technically classified as such. Tomatoes, for example, develop from a single flower with one ovary and contain multiple seeds embedded in fleshy tissue. Pumpkins and cucumbers are considered a type of berry known as a pepo, with a thick rind surrounding soft interior flesh. Citrus fruits are another specialized berry type called a hesperidium, with segmented flesh inside a leathery skin. The botanical category is based on structure, not sweetness, not size, not cultural expectation. It is a matter of how a flower becomes a fruit. When we understand this, a small shift happens in the way we picture a berry bush. Instead of seeing only clusters of color, We can imagine the earlier stage, the delicate flower cups that came before them. Each blossom carried within it a blueprint for fruit formation. After pollination, those blueprints activated. Cells divided, sugars accumulated, pigments formed, layers thickened. The transformation is gradual. A green ovary slowly swells. Chlorophyll fades. Anthocyanin pigments begin to accumulate. Acids shift. Sugars rise. What begins as a firm, pale structure becomes soft and vivid. So even before we talk about nutrition or history, there is a quiet architecture at work. A berry is not simply a small sweet fruit. It is the final stage of a flower's reproductive design. And once that design reaches maturity, it becomes visible in color. When a berry begins to ripen, something subtle changes inside it long before we notice the color shift. The firm green tissue that once felt tight and slightly bitter starts to soften. The chemistry inside the cells begins to move in a different direction. Sugars increase. Acids adjust. The fruit becomes more inviting. At the earliest stage, many developing fruits contain higher levels of organic acids and lower levels of simple sugars. This gives them a sharp or sour taste. As ripening progresses, enzymes inside the fruit convert stored starches into glucose and fructose, two simple sugars that the human body can absorb quickly. The sourness softens as sugar concentration rises. In berries, this transformation can happen over just a few days. A green strawberry gradually turns pale, then pink, then red as sugar content climbs. A blueberry shifts from hard and green to purple, and finally deep blue. These color changes are tied to pigment production, but they are also synchronized with sweetness. The timing matters. Plants do not benefit from having their fruit eaten too early. Seeds inside unripe fruit are often not fully developed. By delaying sweetness, the plant reduces the chance of premature consumption. When ripeness arrives, the fruit signals it clearly. Bright color, softer texture, a gentle scent, higher sugar concentration. For early humans walking through seasonal landscapes, sweetness had meaning. The human tongue contains taste receptors that respond specifically to sugars. When those receptors activate, the brain interprets it as a sign of available energy. Glucose is a primary fuel for cells, especially brain cells. Fructose, found naturally in fruit, can also contribute to energy metabolism once processed in the liver. Compared to tubers like wild roots, which require digging and often cooking, berries are easy to gather. Compared to meat, which demands tools and risk, berries can often be collected quickly and in clusters. They are lower in calories per gram than fatty animal foods, but during peak season, they can appear in abundance. Caloric density helps explain their role. Berries are mostly water, often more than 80% by weight. This means a handful feels substantial, but delivers a modest amount of energy. In a survival context, that can be useful in two ways. First, it provides hydration along with sugar. Second, it allows for quick intake without overwhelming digestion. The sugar concentration in most berries is moderate compared to cultivated tropical fruits. Wild berries tend to be smaller and less sweet than modern selectively bred varieties. Over centuries, humans favored plants with slightly higher sugar levels and larger size, gradually shifting the balance. Still, even in their wild state, berries offered something important: rapid fuel at the right time of year. Seasonality shaped their impact. In many climates, berries appear in late spring and summer. This period often aligns with increased daylight and higher activity levels. Quick sugars would have supported bursts of movement, walking, gathering, building, exploring. At the same time, berries contain fiber, which slows the absorption of sugar into the bloodstream. This creates a steadier release compared to refined sugars. The balance between sugar and fiber is part of what distinguishes whole fruit from isolated sweeteners. In berries, sugar molecules are embedded within plant cell walls. As you chew, those walls break gradually. Digestion unfolds over time. There is also the scent of ripening fruit. As sugars increase, volatile aromatic compounds develop. These small airborne molecules give strawberries their recognizable fragrance and blueberries their subtle sweetness. The human sense of smell detects these compounds even before taste begins. Scent prepares the body for digestion, signaling that energy is near. So sweetness is not random. It is layered. At the plant level, sugar accumulation marks readiness for seed dispersal. At the human level, sweetness activates receptors that evolve to recognize energy-rich foods. At the ecological level, the alignment between ripening and seasonal cycles ensures that fruit is eaten when seeds are viable. When we hold a berry and notice its softness, we are observing a sequence of internal changes that unfolded cell by cell. Enzymes shifted starch to sugar. Pigments replaced chlorophyll. Cell walls loosened. Aromas formed. All of this chemistry happens quietly, guided by genetic instructions inside each plant. And yet, to us, it feels simple. A small fruit turns sweet. We taste it. Energy moves inward. The plant seeds move outward. The exchange is balanced. Long before controlled farming, before irrigation systems and organized fields, human beings relied on careful observation of seasonal change. In that rhythm, berries occupied a predictable window of time. They appeared after flowers faded and before colder weather returned. For groups living as hunter-gatherers, that window mattered. Archaeological research shows that early human diets were diverse and heavily influenced by local ecology. Plant foods often made up a significant portion of daily intake, especially in temperate and tropical regions. While meat provided dense calories and protein, gathering plants required less risk and often less energy expenditure. Berries, in particular, offered a combination of sugars, water, and micronutrients during months when they were available. Evidence of berry consumption appears in multiple forms. In some archaeological sites, charred seeds have been recovered from ancient hearths, suggesting that berries were gathered, eaten, and sometimes processed. At other sites, microscopic plant remains embedded in dental calculus. Hardened plaque on fossilized teeth reveal traces of plant compounds consistent with fruit and berry consumption. These findings indicate that fruit eating is not a recent habit, but part of a long-standing human pattern. Seasonality shaped how berries were used. In many regions, berries ripen in waves. Early summer might bring strawberries or certain small red fruits. Mid to late summer introduces blueberries, blackberries, or raspberries. In northern climates, lingonberries and cloudberries appear toward the end of the growing season. For early populations, this meant periods of relative abundance followed by scarcity. Because berries are perishable, people developed methods to extend their availability. Drying fruit in the sun or over low heat removes moisture, concentrating sugars and slowing microbial growth. Once dried, berries become smaller, darker, and more stable. Some indigenous North American groups combine dried berries with rendered animal fat and dried meat to create nutrient-dense mixtures such as pemmican. In this form, the sugars from berries blended with protein and fat, producing a balanced, portable food that could last through winter months. Compared to tubers like wild yams or roots, berries require no digging tools. Compared to large animal hunts, they involve little danger. However, their caloric density is modest. A cup of fresh blueberries contains roughly 80 calories, largely from natural sugars. In contrast, a similar weight of cooked tubers may provide more sustained energy, and animal fat delivers even greater density. This suggests that berries likely played a complementary role rather than serving as primary caloric staples in many regions. Yet their micronutrient content adds another dimension. Vitamin C, present in varying amounts depending on species, is essential for collagen formation and immune function. Humans cannot synthesize vitamin C internally. During months when leafy greens are scarce and fresh plant material is limited, stored or preserved berries would have helped maintain adequate intake. This is particularly relevant in colder climates, where winter diets might otherwise rely heavily on animal products and preserved grains. The presence of berry seeds in ancient waste deposits and camp areas also points to another pattern: shared gathering. Berry collection can be done by individuals of different ages. It does not require specialized weapons or strength. This may have contributed to social aspects of food gathering, with children and elders participating alongside adults. The act of scanning shrubs and low plants for bright color engages vision, memory, and pattern recognition. From an ecological standpoint, berry plants often thrive in disturbed soils and forest edges. After natural events like fire or clearing, berry shrubs can grow quickly. This would have made them accessible in transitional landscapes. Their growth patterns align with environments that early humans frequently inhabited: open woodlands, grassland margins, riverbanks. The partnership between human foragers and berry plants mirrors the broader ecological relationship between fruiting plants and animals. As humans moved across terrain, seeds passed through the digestive system could be deposited far from the original bush. Some seeds germinate more effectively after passing through an animal gut, where mild abrasion and exposure to digestive enzymes weaken the seed coat. So when we think of early humans reaching for berries, it was not merely a moment of taste. It was part of a wider system. The plant provided sugars and micronutrients at a particular season. The human carried seeds outward. The landscape slowly changed. The exchange was quiet. It did not require tools, only attention. In the stillness of summer, a small fruit ripened, and someone noticed. When a berry shifts from green to red or from pale to deep blue, that visible change is not simply decoration. It marks a chemical transition inside the fruit. Color, in plants, often signals readiness. And in berries, much of that color comes from a group of plant compounds called anthocyanins. Anthocyanins belong to a broader family of molecules known as flavonoids. These compounds are responsible for many of the reds, purples, and blues seen in fruits and flowers. At the cellular level, anthocyanins are stored inside plant cell vacuoles, small compartments that hold pigments dissolved in watery fluid. The exact shade that we see depends on several factors, including the type of anthocyanin present and the acidity of the surrounding environment. In more acidic conditions, anthocyanins tend to appear red. As the environment becomes less acidic, the same molecules can shift toward purple or blue. This is why cranberry juice, which is naturally acidic, appears bright red, while blueberry juice often looks dark purple. If blueberry pigment is diluted in water or exposed to changes in pH, its color can subtly shift. This color flexibility explains why true blue is relatively rare in the plant world. Producing a stable blue pigment requires specific chemical arrangements and sometimes interactions with metal ions or copigments. Fewer flowering plants produce a clear blue hue compared to red or yellow. Blueberries achieve their characteristic appearance partly through pigment chemistry and partly through surface structure. If you examine a blueberry closely, you may notice a faint, pale coating on its skin. This is known as the epicuticular wax, sometimes called the bloom. It forms a thin, powdery layer that covers the fruit surface. This wax scatters light in a way that enhances the berry's blue appearance. If the bloom is rubbed off, the berry often looks darker, closer to deep purple or almost black. The wax coating serves practical purposes beyond appearance. It helps reduce water loss from the fruit. It can protect against ultraviolet radiation from sunlight. It may also create a slight barrier against pathogens. While it does not make the fruit immune to spoilage, it slows certain processes long enough for the berry to complete its role in reproduction. Anthocyanins themselves may provide protection to the plant. These pigments can absorb certain wavelengths of light, helping shield plant tissues from excess radiation. Under cold or stressful conditions, some plants increase anthocyanin production. This response may help manage reactive oxygen species, unstable molecules that can form when plant cells are under environmental stress. So the color of a berry reflects both its readiness to be eaten and its internal defense system. As berries ripen, chlorophyll, the green pigment used in photosynthesis, breaks down. This allows anthocyanins and other pigments to become visible. At the same time, sugar levels increase, and acids may decrease slightly. The fruit softens as enzymes modify cell walls. The plant coordinates these changes so that color, sweetness, and seed maturity align. From a distance, a cluster of ripe berries stands out against green leaves. For animals with color vision, this contrast is effective. Many birds and mammals can distinguish red and blue hues against foliage. The bright coloration acts as a visual signal that the fruit is ready and nutritious. Unripe fruit often remains green, blending into the background and reducing premature consumption. In this way, color functions as communication. A berry does not move. It cannot chase pollinators or seed carriers. Instead, it changes its chemistry until its presence becomes noticeable. The combination of sweetness and color attracts attention. The wax bloom protects just long enough. The seeds inside reach maturity at the same time that the fruit becomes appealing. The relationship between pigment, protection, and perception is layered but steady. Each shift in shade corresponds to a shift in molecular structure. Each deepening color marks an internal transformation. When we see a bowl of mixed berries, strawberries red, blueberries blue, raspberries dark pink, we are looking at different expressions of similar chemical themes. Sugars, acids, pigments, waxes, cell walls. The visual richness comes from chemistry unfolding quietly inside plant cells, and all of it happens without sound. The fruit changes color, the chemistry settles into balance, and something small becomes visible from far away. Think of berries as small, protected packages resting quietly on a branch. From a distance, they look soft and exposed. Their skins are thin. Their flesh is full of juice. Yet before they are picked, they are not unguarded. Much of what gives berries their color and surface texture also functions as a form of protection. Anthocyanins, the pigments responsible for many red, purple, and blue hues, do more than create visual contrast. Inside plant cells, these compounds can absorb certain wavelengths of light, including ultraviolet radiation. Excess ultraviolet exposure can damage cellular structures. By absorbing portions of this light, pigments help shield delicate tissues, especially as fruit ripens and chlorophyll declines. Cold stress also influences pigment production. In some plant species, lower temperatures trigger increased anthocyanin synthesis. This response may help manage reactive oxygen molecules that accumulate under stress. Reactive oxygen species form naturally during metabolism, but under cold or high light conditions, their levels can rise. Anthocyanins can act as antioxidants within plant tissues, helping stabilize these molecules and reduce cellular damage. So when a berry deepens in color during cool nights, part of what you see is a stress response that strengthens the fruit's resilience. The waxy bloom on blueberries and certain other fruits adds another layer of defense. This thin coating, made of complex lipids, forms on the outermost surface of the skin. It repels water, reducing moisture loss through evaporation. It also limits direct contact between the fruit surface and environmental microbes. The bloom scatters light as it reflects off the berry surface, giving blueberries their dusty appearance. If rubbed away, the fruit looks darker and glossier. But while the bloom affects how we see the berry, its primary role is physical protection. It reduces dehydration and helps buffer against sun exposure. Water regulation is essential. Berries contain a high percentage of water, often more than 80% by weight. Maintaining internal moisture keeps cells firm and functional, while seeds continue maturing inside. If too much water escapes, tissues shrink and stress increases. The wax layer slows that process. Pigments and waxes together extend the fruit's viability. They do not stop eventual spoilage, but they delay it. On the plant, this delay matters. A berry must remain intact long enough for animals to notice and consume it. If it decays too quickly, seeds remain clustered near the parent plant. If it lasts just long enough, the likelihood of dispersal increases. Timing becomes central. A berry begins as a flower. After pollination, it develops while still green and firm. During this stage, seeds are immature. The fruit is often less appealing in taste and harder to digest. As seed development completes, pigment levels rise and sugar concentration increases. The berry becomes brighter and sweeter. At the same time, its seeds are ready for travel. This coordination between defense and attraction is subtle but precise. The fruit protects itself from environmental damage during development. Once mature, it shifts toward visibility and softness. It becomes appealing, but only when the internal work is finished. Pest pressures also shape these traits. While berries aim to attract animals that disperse seeds, they must manage insects and microbes that consume tissue without assisting reproduction. Certain phenolic compounds in berries may deter some pests or slow microbial growth. The balance is delicate. Too much defense and the fruit becomes unpalatable. Too little, and it decays prematurely. From a branch, a cluster of berries appears quiet and simple, but each fruit carries layers of chemical strategy: pigments that absorb light, waxes that repel water, compounds that respond to cold, sugars that signal readiness. Protection does not mean permanence. Eventually, every berry softens beyond repair. Microbes colonize, skins break, the fruit collapses. But in the brief window when pigment, sugar, and seed maturity align, the berry is at its most effective. It is protected enough to survive until it is seen, sweet enough to be chosen, and ready enough to send its seeds onward. Now we follow what berries are for in nature. Seed dispersal. A berry offers a simple trade: sugar in exchange for travel. When an animal eats a ripe berry, the fruit's flesh is digested and absorbed. The seeds inside, however, often pass through largely intact. This outcome is not accidental. Over long stretches of evolutionary time, fruiting plants and animals have shaped each other's traits in ways that make this exchange efficient. Seeds are protected by outer coats made of durable plant tissue. These seed coats help them survive physical stress, moisture changes, and in some cases even digestive passage. When a bird swallows a blueberry whole, or a mammal consumes a cluster of blackberries, the fleshy outer portion breaks down quickly in the stomach and intestines. The seeds move through the digestive tract and are later excreted at a new location. This relocation matters. If seeds fall directly beneath the parent plant, they compete for the same light, water, and soil nutrients. They may also attract pests and pathogens that specialize in that species. But if the seeds travel even a short distance, they encounter new soil conditions and reduced competition. The chance of successful germination increases. In many plant species, passage through an animal's digestive system does more than move the seed. It can also improve germination rates. Research reviews have found that in a large proportion of tested species, gut passage either enhances germination or has neutral effects, with improved germination observed in roughly half of the species studied. The digestive process can soften or scarify the seed coat slightly, allowing water to penetrate more easily once the seed reaches soil. The process is quiet and mechanical. Acid in the stomach, mild abrasion in the intestines, then release into soil along with organic waste that provides nutrients. The seed lands with built-in fertilizer. This partnership is known as mutualism. The animal gains energy and hydration. The plant gains dispersal. Neither side consciously negotiates. The exchange is shaped by natural selection over many generations. The visible features of berries reflect this relationship. Bright colors stand out against green leaves, especially to animals with color vision. Birds, for example, can distinguish red and blue hues clearly. The timing of ripening aligns with periods when dispersers are active and mobile. Sugars peak when seeds are mature. Texture also plays a role. Many berries are soft enough to be eaten without much chewing. This reduces the likelihood that seeds are crushed and damaged. Smaller seeds pass through more safely than large, hard pits found in other fruit types. Berry-producing shrubs often grow in transitional or disturbed habitats, forest edges, clearings, riverbanks. These are areas where animals frequently travel. Such locations increase the likelihood that fruit will be encountered and consumed. Over time, this dynamic has shaped plant distribution patterns. A single berry bush can give rise to multiple new plants spread across a landscape. Birds may carry seeds kilometers away. Mammals deposit them in clusters along paths and resting areas. The exchange also influences plant population structure. Genetic diversity increases when seeds spread widely, rather than remaining near the parent. Broader dispersal reduces inbreeding and strengthens resilience across generations. From the perspective of the berry, being eaten is success. The fruit is not the plant's final goal. The seeds are. The flesh is temporary packaging, a vehicle for transport. Once the seeds are delivered to soil, the fruit's role is complete. When we picture a berry bush heavy with ripe fruit, we are looking at a distribution system in motion. Each berry holds potential movement. Each animal that feeds becomes part of the network. There is no noise in this exchange, no visible agreement, just sweetness, just travel, just the steady expansion of plants across time and terrain. And before any of that movement could begin, something else had to happen quietly and precisely. A flower had to be pollinated. Before a berry ever begins to swell, before pigments deepen or sugars rise, there is a brief and delicate stage that determines whether fruit will form at all. It begins with a flower. Berry flowers are often small and understated. A blueberry flower hangs like a tiny bell. A strawberry flower opens with pale white petals around a cluster of yellow structures at its center. Raspberry blossoms are modest, with thin petals and exposed stamens. These flowers do not last long. Their purpose is focused. Inside each flower are male and female structures. The stamens produce pollen, fine grains containing male genetic material. The pistil contains the ovary, which holds the ovules that can become seeds. For fruit to form, pollen must move from the stamen to the receptive surface of the pistil, called the stigma. In some plants, this can happen within the same flower. Self-pollination is possible for certain berry species, but in many cases, cross-pollination, pollen moving between different plants, improves fruit development. This is where insects become important. Bees are among the most effective pollinators of berry plants. As they move from flower to flower, collecting nectar and pollen for their colonies, they inadvertently transfer pollen grains between blossoms. Each visit increases the chance that ovules inside the ovary will be fertilized. Pollination quality affects fruit size and uniformity. Studies have shown that berry fruits receiving adequate bee visitation often grow larger and contain more fully developed seeds. In raspberries and blackberries, fruits pollinated by bees have been observed to be significantly larger, in some contexts roughly 30% larger, than fruits with limited pollination. This difference occurs because each fertilized ovule stimulates surrounding tissue growth. More fertilized ovules mean more evenly developed fruit. Blueberries offer a particularly interesting example. Their flowers are shaped in a way that requires a specific type of pollination, known as buzz pollination. Bumblebees are especially suited for this task. They grasp the flower and vibrate their flight muscles rapidly without flying, shaking loose pollen from specialized anthers. This vibration releases pollen efficiently, allowing it to reach the stigma. Without this mechanical buzzing, some blueberry varieties produce fewer seeds and smaller fruit. The shape of the flower and the behavior of the bee have evolved in coordination. The link between seed number and fruit development is direct. Each seed produces hormones that signal surrounding fruit tissue to expand. If fewer seeds form, growth may be uneven. In strawberries, incomplete pollination can lead to misshapen fruit, where certain sections remain underdeveloped. The outer achenes, each one representing a fertilized ovary, must form evenly for the red receptacle to expand symmetrically. So fruit shape is not random. It reflects the success of pollination. This stage is quiet and brief. Flowering may last only days or weeks depending on climate. During this window, weather conditions matter. Cold temperatures, heavy rain, or lack of pollinators can reduce fruit set. Adequate sunlight, moderate warmth, and active insect populations increase it. From a branch, the change from flower to fruit can seem subtle. Petals fall away. The ovary begins to enlarge. What was once a pale green structure thickens gradually. The transformation from flower to berry is continuous. There is no sudden switch, just steady growth. The entire reproductive cycle depends on this first exchange, pollen reaching stigma. Without it, there is no ovary swelling, no sugar accumulation, no pigment deepening. The bees move quietly between blossoms. The pollen transfers invisibly, and inside each flower, cell division begins. From wild ecosystems to managed fields, this interaction remains essential. Even in commercial berry farms, pollinator presence influences yield. Farmers often introduce managed bee colonies during bloom to ensure consistent fruit development. The sweetness we associate with berries begins here, in a flower cup holding pollen, in the brief meeting of stamen and stigma. And from this fragile stage, the process of fruit formation unfolds, leading eventually to the berries we recognize, harvest, and carry home. The strawberry feels familiar. It appears in desserts, in breakfast bowls, in fields where people bend low with small baskets in hand. Its shape is recognizable. Its color is unmistakable. And yet, the strawberry as we know it is not an ancient fruit in its current form. It is the result of a relatively recent human pairing. Wild strawberries existed long before organized agriculture. Small species of Fragaria grew in forests and meadows across Europe, Asia, and North America. These early fruits were much smaller than modern varieties, often no larger than a fingernail. They were fragrant and sweet, but delicate. In Europe, wild woodland strawberries were cultivated in gardens by the 13th century. These early attempts at cultivation did not dramatically change the fruit's size. They simply brought the plant closer to human settlements. Gardeners selected slightly larger or more flavorful plants, but the transformation remained gradual. The turning point came in the 18th century. Two strawberry species, Fragaria virginiana from North America and Fragaria chiloensis from the Pacific coast of South America, were introduced to Europe. Each species had strengths. F. virginiana produced flavorful fruit. F. chiloensis, especially from Chile, produced larger berries. When these two species were grown near each other in France, natural cross-pollination occurred. The result was a hybrid species known today as Fragaria X ananassa. This hybrid combined desirable traits from both parent species: larger fruit size and improved flavor. It became the foundation for modern garden strawberries. The process did not stop there. Through the 1800s, breeders selected plants with greater yield, hardiness, and consistency. Named varieties began to appear. Each generation of cultivation focused on specific traits: resistance to disease, adaptation to different climates, uniform ripening. Unlike wild berries that spread mainly through seed dispersal, strawberries also propagate through runners. A mother plant sends out horizontal stems along the ground. At intervals, these runners produce new plantlets that root and form independent crowns. This vegetative reproduction allows growers to clone plants with desirable traits. Instead of waiting for seeds to produce variable offspring, farmers can maintain consistent varieties. Modern strawberries are larger, firmer, and more uniform than their wild ancestors. Selective breeding has increased sugar content and extended shelf life. At the same time, this breeding process has altered the balance between size and flavor in some cases. Wild strawberries often contain concentrated aroma, compounds despite their small size. Larger cultivated varieties sometimes trade intensity for volume and transport stability. The strawberry's transformation reflects a broader agricultural pattern. Humans observe variation within a species. They favor certain traits. Over generations, these preferences reshape the plant's characteristics. Yet even as strawberries changed in size and sweetness, their underlying structure remained the same. The fruit still forms from a flower's receptacle. The achene still dot the surface. Pollination still determines shape. The plant still produces runners that extend outward. The modern strawberry is both ancient and new. Ancient in its genus and reproductive design. New in its scale and global reach. Today, strawberries are cultivated across many continents. They are grown in open fields, in greenhouses, even in vertical hydroponic systems. Controlled environments allow year-round production in some regions, but the lineage of these plants traces back to that 18th century hybridization. If you hold a strawberry and look closely, you can still see the evidence of its origins. The seeds on the outside, the central pale core, the way the fruit narrows toward a pointed tip. These features echo the wild ancestors that once grew unnoticed in forest clearings. What changed most dramatically is not the flower's structure, but the human involvement. Through selection and crossing, humans reshaped the fruit scale. From small woodland berries to the larger forms we recognize today. And while strawberries followed one path of domestication, another berry remained stubbornly wild for much longer. That berry was the blueberry. For much of human history, blueberries resisted cultivation. They grew in the wild across North America, in forests, along bog edges, and in sandy, acidic soils. Indigenous communities harvested them for thousands of years. Wild blueberries were eaten fresh, dried for winter storage, and mixed into preserved foods. But attempts to transplant these shrubs into typical garden soil often failed. The reason was hidden beneath the surface. Blueberries belong to the genus Vaccinium, and they have a specific soil requirement that limits where they thrive. They prefer highly acidic soil, typically in the range of pH 4.5 to 5.5. Most agricultural soils are less acidic than this. When planted in neutral or slightly alkaline soil, blueberry plants struggle to absorb nutrients properly, particularly iron. Leaves yellow, growth slows, fruit production declines. For a long time, this requirement made blueberries difficult to farm intentionally. In the early 1900s, a turning point arrived through collaboration between a botanist and a farmer. Dr. Frank Coville, a U.S. Department of Agriculture scientist, studied wild blueberry populations and identified the plant's need for acidic soil. He conducted experiments to determine which environmental conditions were essential for growth and fruiting. At the same time, Elizabeth White, the daughter of a New Jersey cranberry grower, saw the commercial potential of cultivated blueberries. Cranberries also prefer acidic soil, so her family's land was well suited for experimentation. White partnered with Coville, providing access to wild plants and farmland for controlled breeding. Their work focused on selecting wild high-bush blueberry plants with desirable traits, larger fruit size, better flavor, consistent yields, and then propagating them under controlled acidic conditions. They also confirmed that blueberries require a chilling period in winter to break dormancy and flower properly the following season. By 1916, the first commercial crop of cultivated high-bush blueberries was harvested in New Jersey. From that point, breeding programs expanded. Researchers selected plants that could tolerate slightly different climates, extending cultivation beyond the northeastern United States. High-bush blueberries became the dominant commercial variety, but other types exist. Low-bush blueberries, often called wild blueberries, still grow naturally in parts of Canada and the northeastern United States. These smaller berries are harvested from managed wild stands rather than from planted rows. Rabbit-eye blueberries, adapted to warmer climates, are grown in southern regions. Selective breeding gradually increased berry size, improved firmness for transport, and enhanced sweetness. But breeders had to maintain the plant's core requirement: acidity. Modern blueberry farms often amend soil with peat, sulfur, or pine bark to maintain proper pH levels. Irrigation systems deliver water carefully, as blueberry roots are shallow and sensitive to drought. Another important feature of blueberries is their relationship with soil fungi. Blueberry roots form associations with mycorrhizal fungi that help the plant absorb nutrients efficiently in acidic environments. This partnership is part of why blueberries evolved to thrive in soils that would challenge many other crops. So while strawberries adapted readily to gardens centuries ago, blueberries required scientific understanding before large-scale cultivation could succeed. Today, blueberries are grown across North America, South America, Europe, and parts of Asia. Controlled breeding has produced varieties suited to different day lengths and climates. Cold, hardy cultivars withstand northern winters. Southern adapted varieties require fewer chilling hours. Despite these changes, blueberries still follow the same internal pattern described earlier. A flower forms, pollination occurs, the ovary swells, anthocyanins accumulate, the wax bloom develops, sugars rise. The difference is scale. What once grew only in specific acidic patches of wild land now fills carefully managed fields. But even in those fields, the plant remains particular about its environment. The soil must be right. The winter must be cold enough. The pH must remain low. Only then will the small white flowers give way to blue fruit. And once harvested, those berries enter a new stage, one where their protective systems begin to weaken. From cultivation, we move next to fragility, because as carefully as berries are grown, they do not last long after picking. A freshly picked berry looks firm and complete. Its skin is smooth. Its color is deep. Its flesh holds together under gentle pressure. Yet even in this moment of apparent stability, change has already begun. Berries are structurally delicate. Most true berries have thin outer skins and soft internal tissue, made up of loosely packed plant cells filled with water. In many species, water makes up more than 80% of the fruit's weight. This high moisture content gives berries their refreshing quality, but it also makes them vulnerable. The same sugars that attract animals and provide quick energy also create an ideal environment for microorganisms. Yeasts, molds, and bacteria exist everywhere, in soil, in the air, on plant surfaces. While berries are still attached to the plant, their wax bloom and intact skin offer some protection. Once harvested, that protective balance shifts. The epicuticular wax bloom on blueberries and certain other fruits reduces water loss and limits direct contact with microbes. However, handling, washing, and transport can disturb this thin layer. Even small abrasions in the skin create entry points for fungal spores. Because berry tissue is soft, it bruises easily. A bruise occurs when pressure damages internal cells, breaking cell walls and releasing juice. That released moisture provides nutrients for microorganisms. Mold spores that land on the surface can germinate quickly in such conditions. Temperature plays a central role in spoilage speed. At warmer temperatures, microbial growth accelerates. Enzymatic reactions inside the fruit also continue after harvest, gradually breaking down cell walls and softening the structure further. Refrigeration slows both microbial multiplication and internal enzymatic processes. Cold storage does not stop spoilage completely, but it extends shelf life by reducing reaction rates. Different berries spoil at different speeds. Raspberries and blackberries, composed of many small drupelets with tiny gaps between them, tend to deteriorate quickly. Strawberries, with their exposed achenes and delicate flesh, also have limited shelf life. Blueberries often last slightly longer due to their thicker skin and wax bloom, but they too are perishable. Mold growth is often visible as a soft, fuzzy layer, typically white or gray at first, sometimes turning darker as spores mature. The fruit beneath becomes soft and collapses. Because berries are stored in close proximity, mold can spread rapidly from one fruit to another. The fragility of berries is directly linked to the same traits that made them effective for seed dispersal. Thin skins make them easy to eat. High sugar content attracts animals. Soft flesh allows seeds to pass through digestive systems without damage. These characteristics, beneficial in nature, reduce durability after harvest. Water loss also contributes to spoilage. Even in cold storage, berries slowly lose moisture through their skins. As water evaporates, cells shrink and the fruit becomes wrinkled. Texture changes before visible mold appears. Mechanical damage during transport compounds these effects. Stacked containers, vibration during shipping, and temperature fluctuations all influence longevity. Modern supply chains use careful packaging, rapid cooling, and controlled humidity to extend shelf life. Yet compared to many other fruits, berries remain short-lived. The speed of spoilage has shaped human behavior for centuries. Because berries do not store well in their fresh state, people developed preservation methods. Drying removes water, inhibiting microbial growth. Cooking berries with sugar to create jam increases osmotic pressure, making the environment less favorable for bacteria. Freezing slows enzymatic reactions dramatically, allowing berries to be stored for months. The softness of a berry is part of its appeal. When eaten fresh, it requires little effort to bite through. The texture collapses gently, but that same softness signals that the fruit's tissues are not built for long endurance once separated from the plant. From flower to ripening fruit to fragile harvest, the life cycle moves steadily forward. On the branch, pigments and waxes buy time. After picking, time becomes limited, and as spoilage sets in, humans turn toward methods that extend the season, preserving sweetness long after the bushes have emptied. Imagine hands spreading freshly gathered berries across a flat surface in the sun. The fruit is plump and bright at first. A shallow basket is tipped gently. The berries roll outward in a single layer. Air moves across them. Heat begins to draw moisture from their skins. For hunter-gatherer communities, this simple act was part of a seasonal rhythm. Summer brought abundance. Bushes filled with ripe fruit for a few short weeks. But winter would come, and with it, scarcity. The strategy was not to eat only for the moment, but to extend the season. Drying was one of the oldest preservation methods. By reducing water content, people slowed microbial growth. Water is essential for bacteria and molds to multiply. When berries dry, their internal moisture drops, and the environment becomes less favorable for spoilage organisms. As water leaves, the berry changes visibly. It shrinks, the skin wrinkles, the color darkens. Sugars become more concentrated. What was once soft and fragile becomes leathery and dense. The flavor intensifies because the same amount of sugar is now held in a smaller volume. This process does not eliminate nutrients. While some vitamin C is lost during drying due to exposure to air and light, dried berries can still retain meaningful amounts, especially if dried gently and stored carefully. In climates where fresh produce disappears for months, dried berries carried nutrients into winter. Freezing, a method now common, mimics natural winter preservation. In cold climates, berries left outdoors during early frosts could freeze naturally. Freezing slows enzymatic activity and microbial growth dramatically. Modern freezers allow berries to be preserved close to their fresh state, retaining much of their vitamin C and fiber. Indigenous preservation practices across North America and other regions demonstrate how berries were integrated into long-term food planning. Dried berries were often mixed with other preserved foods. In some traditions, dried berries were combined with rendered animal fat and dried meat to form pemmican. This mixture provided a balanced source of energy: carbohydrates from fruit, protein from meat, and fat for sustained calories. Pemmican could be stored for extended periods and transported over long distances. In northern regions, berry preserves were also prepared by gently heating fruit and storing it in containers where natural sugars acted as partial preservatives. These practices varied by culture and climate, but shared a common goal: carry summer forward. Preservation can be understood as time-shifting nutrients. The berry remains the same plant product, but its season changes. A fruit gathered under bright sunlight becomes part of a winter meal months later. The sugars and fiber that once supported activity in warm weather become part of nourishment during shorter days. This strategy reduced dependence on any single food source. In environments where plant growth pauses during cold months, stored berries offered diversity in taste and nutrition. Even modest amounts of preserved fruit could contribute vitamin C when fresh greens were unavailable. Drying also made berries lighter and easier to transport. A handful of dried berries weighs less than the same quantity fresh because water has been removed. For mobile groups, reducing weight while preserving calories mattered. The visual shift from plump to wrinkled marks the transition. The berry loses volume but gains durability. Its surface tightens. Its color deepens toward dark red or nearly black. The transformation is practical, not decorative. This process reflects careful observation. Early communities learned when berries were at peak ripeness. They understood that drying worked best when fruit was neither underripe nor overripe. They selected days with sun and airflow. Preservation is not just about preventing spoilage. It is about extending access to nutrients across time. The same berry that would have decayed within days on a kitchen counter could last months when dried properly. Over generations, these practices became embedded in daily life. The act of collecting, drying, storing, and later consuming berries followed seasonal cycles. And as preservation techniques evolved, they became more than survival tools. They became part of culture. Jars lined shelves. recipes were shared. Traditions formed around fruit transformed by time. The sound begins quietly. A pot rests over steady heat. Fresh berries collapse as they warm, their skins split. Juice releases. The mixture thickens slowly as steam rises into the air. This is another form of preservation, not through drying alone, but through concentration and sugar. As berries cook, water evaporates. Natural sugars become more concentrated. When additional sugar is added, the environment inside the pot changes further. High sugar concentrations create osmotic pressure that makes it difficult for many bacteria and molds to grow. Microorganisms require available water to multiply. Sugar binds free water molecules, reducing what scientists call water activity. Lower water activity slows spoilage. In this way, preservation becomes chemistry. Berry preserves have a long history in parts of Europe. By the Renaissance era, fruit cooked with sugar was considered a valued preparation. In earlier centuries, sugar itself was rare and expensive in many regions. It arrived through trade networks and colonial expansion. When sugar became more available after the 1500s, fruit preserves became more common across households. Cooking fruit with sugar does more than extend storage. Heat breaks down plant cell walls, especially pectin, a natural fiber present in berry skins and flesh. Pectin forms a gel structure when heated with sugar and acid. This creates the thick, spreadable texture associated with jam. Different berries contain different natural pectin levels. Strawberries are relatively low in pectin, so traditional recipes often combine them with added pectin or with fruits naturally higher in it. Raspberries and blackberries contain moderate amounts. The balance between fruit acidity, sugar concentration, and pectin determines how firmly a jam sets. Preserves appear under many names depending on region and preparation style. In France, confiture refers to fruit cooked with sugar until thickened. In Eastern Europe and parts of Russia, Varenier describes whole fruits suspended in syrup. Strawberry jam became widespread in England and other European countries as sugar imports increased. Mixed berry preserves emerged as seasonal blends when multiple fruits ripened at the same time. Jars lined with deep red, purple, and dark blue contents represent stored fruit in concentrated form. Each jar holds fruit from a specific harvest season. The process of sealing hot preserves into sterilized containers creates a vacuum as they cool. This reduces exposure to air and further slows microbial growth. Preservation in this context is not simply sweetness added for taste. It is a storage technology. Sugar and heat together create conditions that extend shelf life significantly compared to fresh fruit. Over time, berry preserves moved from luxury items to common household staples. In colder climates, they provided variety in winter meals. A spoonful of jam added fruit flavor to bread or porridge long after the growing season had ended. Unlike dried berries, preserves maintain a softer texture. The fruit structure breaks down during cooking, forming a unified spread rather than individual pieces. The color deepens under heat as pigments interact and concentrate. There is also a shift in scale. Drying and mixing berries were practices used by mobile groups and small communities. Jam making often became associated with settled agricultural life. It required access to cooking vessels, storage jars, and consistent sugar supplies. Yet both drying and jam making share the same underlying goal: extend the usefulness of a perishable fruit. In the pot, the transformation is visible. Whole berries soften and collapse. Bubbles form and burst at the surface. The liquid thickens gradually. When cooled, it holds shape instead of flowing freely. This transformation marks a shift from fresh to preserved. The berry remains recognizable in flavor, but its structure has changed. It is no longer fragile and short-lived in the same way. It has been prepared to last. From temperate kitchens and Renaissance preserves, we move northward now, to regions where berries were not simply seasonal treats, but essential winter foods. In those places, preservation was not optional. It was necessary. In northern regions, summer arrives quickly and leaves just as fast. The growing season is short. The sun stays high for long hours, and plants move rapidly through flowering and fruiting. In these brief windows, berries become especially important. Across Scandinavia, parts of Russia, Canada, and Arctic-adjacent landscapes, wild berries are woven into daily life and cultural identity. The terrain in many of these areas supports low-growing shrubs that thrive in cool climates and acidic soils. Lingonberries, cloudberries, bilberries, and crowberries dot the ground in late summer. Lingonberries grow close to the forest floor, small and bright red against green leaves. Their flavor is tart rather than intensely sweet. In Scandinavian countries, lingonberry jam is a common staple. It is traditionally served alongside savory foods such as meat dishes and potatoes. The pairing may seem unusual at first, but the acidity of the berries balances rich flavors. Over generations, this combination became part of everyday meals. Cloudberries are especially valued in Arctic and subarctic regions. These berries grow in marshy tundra and peatlands. When ripe, they turn a deep golden orange. Their harvest window is brief, and yields can vary from year to year depending on weather conditions. In some regions, cloudberries are referred to as Arctic gold due to their rarity and importance. They are eaten fresh, made into jams, or mixed with cream and desserts. In northern climates, preservation is not simply tradition, it is necessity. Fresh produce becomes scarce during long winters. Historically, people relied on stored grains, dried fish, preserved meats, and whatever plant foods could be kept through cold months. Berries, when dried or turned into preserves, provided one of the few reliable plant-based sources of vitamin C. Vitamin C deficiency leads to scurvy, a condition caused by impaired collagen production. In extreme cases, scurvy results in weakened connective tissue, bleeding gums, and poor wound healing. Populations in northern environments learned through experience which wild foods prevented these symptoms. Lingonberries, cloudberries, and rosehips were valued not only for flavor, but for their protective nutritional properties. Drying berries allowed families to store fruit in small containers for winter use. Jam making became common once sugar supplies were accessible. Even modest quantities of preserved berries could supplement winter diets. The landscape itself shaped these practices. In Arctic and subarctic areas, berry shrubs grow low to the ground, close to mosses and lichens. During harvest season, fields and forests appear dotted with bright color just above the soil surface. Gathering requires attention and patience. The fruit may be small, but it appears in clusters. Because berries thrive in poor, acidic soils that are unsuitable for many crops, they became reliable foods in regions where agriculture is limited. Unlike wheat or corn, berry shrubs require no plowing or sowing. They return each year on their own. The cultural role of berries extends beyond nutrition. Harvest season often involves family outings, shared gathering, and community exchange. Recipes pass through generations. Certain berries become associated with local identity. In some countries, laws allow public access to wild berry patches, reinforcing their place in national life. In these northern settings, berries are not treated as occasional luxuries. They are seasonal anchors. Their appearance signals the height of summer. Their preservation signals preparation for winter. The bright red of lingonberries or the golden hue of cloudberries stands out sharply against muted tundra and forest floors. The contrast between landscape and fruit reinforces their visibility, much like in earlier discussions of color signaling for seed dispersal. Here, however, the relationship is human-centered. The fruit supports survival through extended cold periods. Preservation methods ensure that vitamin C and other nutrients remain available when fresh greens vanish. From this cultural and geographic context, we shift now to one specific nutrient that made berries especially valuable in winter: vitamin C, a simple molecule with a crucial role in human biology. Vitamin C is a small molecule with a large role. Its chemical name is ascorbic acid. Humans cannot produce it internally. Unlike many animals that synthesize vitamin C in their own bodies, humans must obtain it from food. Without regular intake, deficiency develops over time. Vitamin C is essential for collagen production. Collagen is a structural protein that forms connective tissue throughout the body. In skin, blood vessels, tendons, ligaments, and bone. The body uses vitamin C as a cofactor in the enzymatic reactions that stabilize collagen fibers. Without sufficient vitamin C, collagen becomes weak. Blood vessels become fragile. Wounds heal poorly. This deficiency condition is known as scurvy. Historically, scurvy appeared most often in situations where fresh plant foods were absent for extended periods. Long sea voyages and harsh winters created risk. Symptoms include fatigue, swollen gums, joint pain, and in severe cases, bleeding and infection. Berries became important in regions where other fresh produce was limited. Many berry species contain measurable amounts of vitamin C, though levels vary by type. Strawberries are particularly high in vitamin C. A single cup of fresh strawberries can provide approximately 150 milligrams of vitamin C, exceeding the average adult daily requirement. Raspberries contain around 46 milligrams per cup. Blueberries contain less, roughly 10 milligrams per cup, while cranberries provide about 14 milligrams per cup in fresh form. These values vary depending on growing conditions, ripeness, and storage, but they illustrate an important pattern. Certain berries are dense sources of vitamin C relative to their size. In northern climates, wild berries such as lingonberries and cloudberries were traditionally valued for this reason. Rose hips, the fruit of wild rose plants, are especially high in vitamin C and were also used as winter remedies in parts of Europe. When dried or made into syrups, these fruits help prevent deficiency during months without fresh greens. The role of vitamin C extends beyond scurvy prevention. It functions as an antioxidant, helping neutralize reactive oxygen species in human cells. It supports immune function and aids in iron absorption from plant-based foods. But its most historically visible function was preventing connective tissue breakdown. In environments where diet shifted toward preserved meats and grains during winter, vitamin C-rich berries acted as nutritional insurance. Even small amounts consumed periodically could maintain adequate levels. Preservation methods influence vitamin C retention. Heat and prolonged exposure to air degrade ascorbic acid over time. Drying under low heat preserves more vitamin C than prolonged boiling. Freezing retains much of the vitamin C content, especially when berries are frozen soon after harvest. The concept of berries as winter insurance is rooted in practical observation. Communities that included preserved berries in winter diets were less likely to develop deficiency symptoms. Knowledge of this relationship was passed through experience long before the underlying chemistry was understood. Today, vitamin C deficiency is rare in most developed regions due to year-round access to fruits and vegetables. Yet the historical importance of berries remains part of nutritional understanding. It is notable that a small fruit growing close to the ground could influence survival in extreme climates. The concentration of vitamin C in certain berries makes them disproportionately significant relative to their size. From a biological perspective, vitamin C supports the maintenance of tissues that hold the body together. From a historical perspective, berries supported populations through long winters when fresh produce was unavailable. A cup of strawberries delivers more than sweetness. It delivers a molecule required for structural integrity. A handful of preserved lingonberries or cloudberries once carried protection across months of cold. Vitamin C is only one component of berries, but it is one with a clear and measurable role. And while vitamin C acts at the molecular level, another component of berries works at a slower structural pace within the digestive system. That component is fiber, the quiet structure inside a soft fruit. When you bite into a berry, the first sensation is sweetness and juice. But not everything dissolves. Inside the soft flesh are structures that resist digestion. Tiny seeds, thin skins, strands of plant cell wall material. These components are collectively known as dietary fiber. Fiber is the portion of plant food that human digestive enzymes cannot fully break down. Many berries are notable for their fiber content relative to their size. Raspberries contain approximately 8 grams of fiber per cup. Blueberries provide around 3.6 grams per cup. Strawberries contain about 3 grams per cup. These values vary slightly depending on growing conditions and measurement methods, but the pattern is consistent. Berries are fiber-dense compared to many other fruits. Fiber exists in different forms. Some fiber is soluble, meaning it dissolves in water and forms a gel-like substance. Pectin, found in berry skins and flesh, is one example. Other fiber is insoluble, meaning it does not dissolve and instead adds bulk to stool. The tiny seeds and structural plant fibers in berries contribute to this insoluble fraction. When berries are eaten whole, their fiber slows digestion. Sugars are not absorbed instantly. Instead, they move through the digestive tract embedded within plant tissue. The gel-like quality of soluble fiber can slow the movement of food through the stomach and small intestine. This gradual process leads to steadier absorption of glucose into the bloodstream. This is one reason why whole berries tend to have a relatively moderate glycemic impact compared to refined sugars. The combination of fiber, organic acids, and natural plant compounds influences how quickly blood sugar rises after consumption. Rather than a sharp spike, the response is often more gradual. In addition to influencing blood glucose patterns, fiber supports stool formation. Insoluble fiber adds bulk, helping maintain regular bowel movements. Soluble fiber absorbs water, contributing to stool softness. Together, these effects support digestive function. The structure of a berry reflects this fiber content. If you examine a raspberry closely, you can see the many small drupelets and the seeds within them. Each tiny seed is surrounded by fibrous plant tissue. Blueberries, though smoother on the outside, also contain small seeds and skin structures that contribute to fiber intake. Cooking berries, as in jam preparation, breaks down some structural components, but does not eliminate fiber entirely. Drying berries concentrates fiber along with sugars, since water is removed but structural material remains. Fiber is not absorbed into the bloodstream in the way that sugars and vitamins are. Instead, it continues moving through the digestive tract. In the large intestine, some types of fiber encounter another system, one that cannot be seen directly. That system is the microbiome. Inside the colon live trillions of microorganisms. These microbes interact with dietary fiber in ways that influence overall health. Certain fibers serve as substrates for fermentation by beneficial bacteria. This fermentation produces short-chain fatty acids, molecules that support colon cell health. But even before reaching the colon, fiber has already shaped digestion. It slows transit time in the upper gastrointestinal tract. It moderates appetite signals. It influences nutrient absorption rates. The softness of a berry can make it seem simple and quickly consumed. Yet within that soft texture lies structural complexity. Fiber is part of what gives berries their internal framework. Without it, the fruit would collapse more easily. With it, digestion becomes steadier. In earlier sections, we looked at sugars rising during ripening. Now we see the counterbalance. Sugar provides quick energy. Fiber slows its release. Seeds and skins that pass through digestion intact serve another ecological purpose. They may still be capable of germination if dispersed into soil. In that sense, fiber supports both plant reproduction and human digestive function. From the sweetness that dissolves to the structure that remains, berries offer multiple layers of interaction within the body. And once fiber reaches the large intestine, its role changes again. From structure in the mouth to fermentation in the microbiome, a quiet transformation continues beyond taste. After a berry is swallowed, digestion continues beyond what can be felt. Sugars are absorbed in the small intestine. Vitamins enter the bloodstream. But fiber and certain plant compounds move onward into the large intestine. There, another ecosystem waits. The human colon contains trillions of microorganisms, bacteria, archaea, and other microscopic life forms collectively known as the gut microbiome. These microbes interact with dietary components that human enzymes cannot fully digest. Berry fibers, including pectins and other complex carbohydrates, serve as substrates for microbial fermentation. When bacteria ferment these fibers, they break them down into smaller molecules called short-chain fatty acids, or SCFAs. The primary SCFAs produced are acetate, propionate, and butyrate. Butyrate is particularly important for colon health. It serves as a primary energy source for colon cells, known as colonocytes. Adequate butyrate production supports the integrity of the intestinal lining. A healthy gut barrier prevents unwanted substances from passing into the bloodstream while allowing nutrients to be absorbed. Acetate and propionate also contribute to metabolic signaling and energy balance. These molecules enter circulation and influence processes in the liver and other tissues. While research continues to explore the full range of effects, SCFAs are generally associated with positive gut and metabolic outcomes when produced in balanced amounts. Berries contribute not only fiber, but also polyphenols, plant compounds including flavonoids and anthocyanins. Some of these polyphenols reach the colon largely intact. There, microbes metabolize them into smaller bioactive compounds. This interaction can influence microbial composition. Research summaries suggest that berry consumption may promote beneficial bacterial populations while suppressing certain less favorable strains. The term prebiotic refers to substances that stimulate the growth or activity of beneficial microorganisms. Berry fibers and polyphenols can act in this prebiotic role. The fermentation process itself is quiet and continuous. There is no conscious sensation when bacteria break down fiber into SCFAs, yet this process influences gut environment over time. Another effect of fiber is its influence on blood glucose stability. As mentioned earlier, fiber slows the absorption of sugars in the small intestine. When berries are consumed as whole fruits rather than as juices or refined extracts, the presence of fiber reduces rapid spikes in blood sugar. Over repeated meals, steadier glucose patterns may contribute to metabolic balance. The microbiome interacts with this pattern as well. SCFAs produced during fermentation can influence hormones related to appetite and insulin sensitivity. While these effects are complex and still being studied, the overall pattern suggests that whole berries contribute to digestive stability through multiple pathways. The balance between sweetness and structure becomes important again. Without fiber, sugar would move rapidly into the bloodstream. Without polyphenols, the microbial interaction would differ. The intact fruit delivers both. In addition to fiber and polyphenols, berries contain organic acids that influence digestion. Acidity may affect how quickly the stomach empties and how enzymes interact with food. These effects are subtle, but contribute to the overall digestive experience. The microbiome is dynamic. Its composition changes based on diet, environment, age, and other factors. Regular intake of fiber-rich foods, including berries, supports microbial diversity. Greater diversity is generally associated with resilience in the gut ecosystem. It is important to note that individual responses vary. Different people host different microbial communities. The same food can produce slightly different fermentation profiles depending on the existing microbiome. Still, the consistent element is this: fiber does not disappear unused. It becomes fuel for another system. The visible berry becomes invisible metabolites. The sweetness dissolves. The structure remains. Microbes ferment quietly. Short-chain fatty acids form. Colon cells use them. Signals move outward into the body. From a small fruit, a cascade of interactions unfolds beyond awareness. And from this digestive stability, researchers have begun examining another area of interest: the relationship between berry compounds and inflammation throughout the body. A shift from the gut to broader physiological signals. Berries are often described with one word: antioxidants. The label appears on packaging, in articles, and in headlines. The term refers to compounds that can neutralize reactive oxygen molecules, sometimes called free radicals, in laboratory settings. Reactive oxygen species form naturally in cells during metabolism. In excess, they can damage proteins, lipids, and DNA. Antioxidants can stabilize these molecules by donating electrons, reducing potential harm. Many berries contain anthocyanins and other polyphenols that show antioxidant activity in test tubes. Blueberries, blackberries, raspberries, cranberries all contain varying combinations of these compounds. Anthocyanins give berries their deep reds, purples, and blues. Other polyphenols contribute additional chemical diversity. For many years, researchers measured antioxidant capacity using a laboratory test called ORAC, Oxygen Radical Absorbance Capacity. This method evaluated how well a food extract could neutralize a specific reactive molecule under controlled conditions. Berries frequently ranked high in ORAC testing compared to many other fruits. Blueberries and blackberries often appeared near the top of these rankings. In one comparative study frequently cited in summaries, blueberries demonstrated among the highest antioxidant capacities of commonly consumed berries when measured using ORAC methodology. However, it is important to understand what ORAC scores represent and what they do not. ORAC tests measure antioxidant activity in vitro, meaning outside the human body, under controlled laboratory conditions. The human body is far more complex. After ingestion, polyphenols are absorbed, metabolized, transformed by gut bacteria, and distributed in different tissues. The concentration that reaches specific cells may differ significantly from what is observed in test tubes. Because of these limitations, the United States Department of Agriculture eventually removed its ORAC database from public resources, noting that ORAC values do not directly translate into health effects in humans. Still, the laboratory findings sparked further investigation. Beyond direct antioxidant activity, polyphenols may influence cellular signaling pathways related to inflammation. Rather than acting solely as chemical scavengers of free radicals, these compounds can affect how cells respond to stress. Anti-inflammatory effects are often described in simplified terms as lowering certain inflammation signals. In laboratory and animal models, Berry extracts have been shown to influence markers associated with inflammatory pathways. These effects vary by dose, compound type, and experimental conditions. Human studies examining inflammatory markers after berry consumption show mixed but sometimes promising results. Some trials report modest reductions in specific inflammatory biomarkers following regular intake of berry-rich diets. Others find minimal change. Differences in study design, participant health status, and berry preparation contribute to variation. What remains consistent is that berries contain biologically active compounds beyond basic vitamins and fiber. Anthocyanins, flavonols, tannins, and other polyphenols interact with cellular systems in ways still being mapped. The antioxidant label, while simplified, reflects real chemical properties observed in laboratory testing. But translating those properties into clear human outcomes requires careful research. Instead of thinking of berries as single-function antioxidant bombs, it is more accurate to view them as complex chemical packages. Their compounds participate in multiple pathways. They interact with gut microbes. They influence signaling molecules. They may contribute to overall dietary patterns associated with lower chronic disease risk. The key word is association. Populations consuming diets rich in fruits and vegetables, including berries, often show lower rates of certain chronic conditions. But isolating one compound or one fruit as the sole cause is difficult. The science continues to unfold. Anthocyanins show antioxidant potential in vitro. Polyphenols interact with inflammation pathways. ORAC scores highlighted berries as chemically active foods. But the full story within the human body remains an area of ongoing research. One of the most intriguing areas of study moves from cellular oxidation towards something more measurable in daily life. Memory, attention, processing speed, the brain. From chemical reactions to cognitive function, berries have entered another field of investigation. When researchers began looking beyond laboratory antioxidant tests, one of the areas that drew attention was the brain. Cognitive function, including memory, attention, and processing speed, changes gradually over time. In aging populations, researchers study whether certain dietary patterns might influence the rate of decline. Berries entered this conversation because of their flavonoid content, especially anthocyanins. Several human trials have examined blueberry supplementation in older adults. In randomized controlled studies, participants were assigned either a blueberry supplement, often in the form of freeze-dried blueberry powder, or a control product without active berry compounds. Over periods ranging from several weeks to several months, researchers measured cognitive performance using standardized tests. In one study frequently cited in research summaries, older adults with mild cognitive decline consumed blueberry powder daily for 6 months. Compared to controls, participants in the blueberry group showed improvements in certain cognitive measures, including processing speed. Processing speed refers to how quickly the brain can interpret and respond to information. Other trials have reported improvements in specific memory tasks. In some cases, visuospatial memory, the ability to remember visual patterns and spatial relationships, improved after regular blueberry intake. These effects were modest but measurable. It is important to frame these findings carefully. These studies typically involve small sample sizes. Results vary depending on dosage, duration, participant health status, and testing methods. Improvements are often specific to certain tasks, rather than broad across all cognitive domains. The proposed mechanisms involve several pathways. Flavonoids may enhance cerebral blood flow. Imaging studies suggest that flavonoid-rich foods can increase blood flow to certain brain regions temporarily. Improved blood flow supports oxygen and nutrient delivery, which may influence cognitive performance. Polyphenols may also interact with signaling pathways related to neuroinflammation and neuronal plasticity. In laboratory and animal models, certain flavonoids appear to influence pathways involved in learning and memory. Translating these findings into consistent human outcomes remains an ongoing process. In addition to blueberries, studies have examined other berries such as strawberries. Some observational research suggests that higher long-term intake of berry flavonoids is associated with slower rates of cognitive decline in aging populations. Observational studies, however, cannot prove cause and effect. They identify patterns that require controlled trials for confirmation. The timing of intake also matters. Some acute studies measure cognitive performance a few hours after consuming berry products, observing temporary changes in attention or task accuracy. These short-term effects may relate to immediate changes in blood flow or glucose availability. Longer-term trials examine whether repeated intake produces sustained differences. The cautious interpretation is this. Research suggests that berry consumption, particularly blueberries, may support certain aspects of cognitive function in older adults. The evidence is promising but not definitive. Larger and longer trials are needed to clarify the magnitude and consistency of effects. The improvements observed in trials are not dramatic reversals of cognitive decline. They are typically modest enhancements in specific tasks compared to control groups. Still, the consistency of interest in this area reflects something notable. A small fruit rich in flavonoids has become part of scientific inquiry into brain aging. From the field to the laboratory to the clinic, berries continue to be studied not only for nutrient content, but for functional outcomes. It is also worth remembering that these studies examine whole food compounds interacting within complex human systems. The brain is influenced by many factors: genetics, physical activity, sleep, social interaction, overall diet. Berries are one component within that broader picture. The movement from ORAC scores to cognitive trials reflects a shift in research focus, from measuring chemical potential in test tubes to observing functional outcomes in people. The science is still unfolding. The conclusions remain measured, but the question has expanded. From antioxidant capacity to attention and recall, from pigment chemistry to processing speed. And long before clinical trials existed, berries were used in traditional remedies for other purposes, some supported by modern data, others more debated. We move now from modern research labs back into older medicine cabinets and cultural uses, where berries were valued for reasons both practical and symbolic. Before clinical trials and laboratory measurements, berries were part of home remedies and traditional medicine systems. Elderberry is one of the most widely cited examples. The plant Sambucus nigra grows in parts of Europe and North America. Its dark purple berries have been used for centuries in syrups and decoctions. Historical records note that elderberry preparations were used to address symptoms of colds and flu-like illnesses. Hippocrates, writing around 400 BC, is often quoted as referring to the elder tree as a kind of medicine chest, reflecting its broad traditional use. Modern research has examined elderberry extracts for potential effects on viral symptoms. Some small clinical trials suggest that elderberry syrup may modestly reduce the duration or severity of cold and influenza symptoms when taken early. However, sample sizes are often limited, and study designs vary. While results are encouraging in certain contexts, larger trials are needed to establish clear clinical recommendations. Cranberries provide another example where traditional use intersects with modern research. Indigenous peoples in North America use cranberries both as food and for medicinal purposes. In contemporary research, cranberries have been studied for their role in urinary tract health. Certain compounds in cranberries, known as proanthocyanidins, appear to reduce the ability of Escherichia coli bacteria to adhere to the lining of the urinary tract. Adhesion is an early step in infection development. By interfering with this attachment process, cranberry products may reduce the risk of recurrent urinary tract infections in some populations. Meta-analyses examining cranberry juice or supplements suggest a modest reduction in recurrent UTIs, particularly in women with a history of frequent infections. The effect is not universal, and cranberry products are not considered a treatment for active infections. Instead, they may function as a preventive measure for certain individuals. Dosage, formulation, and consistency of intake all influence outcomes. Bilberries, closely related to blueberries, are often associated with a popular historical story. During World War II, British pilots were said to have consumed bilberry jam to improve night vision. This narrative has persisted in popular culture. However, controlled clinical studies have not found strong evidence supporting significant night vision enhancement from bilberry consumption. While bilberries contain anthocyanins similar to blueberries, the night vision claim appears to be more myth than established fact. This contrast between folklore and data provides an opportunity for careful framing. Traditional use indicates long-standing cultural value. Modern clinical research evaluates specific outcomes under controlled conditions. Sometimes the two align, sometimes they do not. Lingonberries in Nordic regions have also been used both as food and as part of herbal traditions. These berries contain natural benzoic acid, a compound that contributes to their preservative qualities. The presence of benzoic acid may partly explain why lingonberry preserves can remain stable with relatively less added sugar compared to some other fruits. While lingonberries have been used in teas and remedies, most evidence supports their value primarily as nutrient-rich foods, rather than as standalone medicinal treatments. Across these examples, a consistent theme appears. Berries have long been part of human attempts to manage health. Some traditional uses now have partial support from modern research. Others remain largely anecdotal. The scientific process requires controlled trials, replication, and careful interpretation. It is also important to distinguish between whole berries consumed as part of a balanced diet and concentrated extracts used therapeutically. Effects observed with standardized extracts may not translate directly to everyday dietary amounts. Traditional medicine systems often integrate berries within broader dietary and lifestyle practices. The fruit becomes one element within a holistic framework. Measured framing helps maintain clarity. Elderberry shows promise in certain symptom contexts but requires further study. Cranberry products may modestly reduce recurrent UTIs in specific groups. Bilberry's night vision reputation lacks strong clinical backing. Lingonberries contain natural preservative compounds and are nutritionally valuable. The conversation remains ongoing. Research continues. Traditional knowledge persists. From remedies and medicine cabinets, we widen our view again, stepping back into symbolism, art, and seasonal meaning. Because berries have not only nourished bodies, they have also shaped stories and rituals. Berries are small, but they appear often in stories. Across cultures, they show up in rituals, decorations, seasonal festivals, and art. Their bright color against green leaves or white snow makes them visually striking. Their brief season gives them symbolic weight. In many European traditions, red berries were associated with late summer and early autumn harvests. Wreaths and garlands sometimes included berry branches as symbols of abundance. The fruit represented the success of the growing season, proof that flowers had been pollinated and that the land had produced food. In colder climates, berries also became symbols of endurance. When the landscape shifted toward winter, preserved berries carried summer forward. The contrast between red fruit and snow appears frequently in visual motifs, paintings, textiles, and seasonal decorations. The color red against white became a seasonal marker. In Nordic regions, berry gathering remains part of cultural identity. Seasonal harvests bring families outdoors. Recipes passed down through generations preserve flavor and memory. Lingonberries, cloudberries, bilberries, each has regional significance. Their use in sauces, desserts, and even spirits reflects a continuity of tradition. Beyond Europe, berries appear in indigenous traditions in North America. Certain berries are linked to seasonal ceremonies or community gatherings. Harvest periods are tied to ecological knowledge, understanding when fruit is ready and how to gather it sustainably. The repetition of berry imagery in art reflects more than nutrition. Berries are visually simple forms, round shapes, clustered, often glossy. They lend themselves to decorative patterns. Medieval manuscripts sometimes included stylized berry vines along margins. Folk art often features berry branches intertwined with leaves. Symbolically, berries can represent fertility because they contain multiple seeds. A single fruit holds potential for many future plants. This quality made them fitting symbols in seasonal celebrations connected to growth and renewal. At the same time, berries carry associations with transience. Their ripening window is brief. A bush that appears full one week may be empty the next. This short season reinforces their connection to cycles: growth, harvest, preservation, dormancy. In literature, berries sometimes appear as markers of place. A character picking berries suggests a rural or woodland setting. The act is quiet and focused. It requires attention to the ground, to leaves, to subtle shifts in color. Culinary traditions continue to evolve while drawing from older patterns. In Scandinavian cuisine, berry sauces accompany savory dishes. In Eastern Europe, varenje and fruit preserves remain common. In North America, berry pies and jams are seasonal staples. Modern chefs incorporate berries into reductions, vinaigrettes, and fermented preparations. Spirits infused with berries, such as certain Nordic liqueurs, extend preservation traditions into contemporary forms. Fermentation and distillation add new layers to long-standing harvest practices. Even today, berry festivals take place in regions where certain fruits define local identity. Blueberry festivals, strawberry fairs, cloudberry celebrations. These events link agriculture, tourism, and cultural memory. The recurring image is simple: a cluster of small red or blue spheres against green leaves, a bowl of mixed berries on a table, a jar of deep purple preserve lined up on a shelf. Each image connects biology and culture. The fruit developed through pollination, pigment formation, seed maturation. Humans gathered it, preserved it, named it, celebrated it. Berries occupy a space where ecology meets ritual. They are products of natural systems, yet they carry symbolic meaning shaped by human interpretation. The repetition of berry imagery across seasons and regions reinforces their place in collective memory. Their small size makes them accessible. Their color makes them noticeable. Their seasonality makes them anticipated. In stories, berries are rarely grand or dramatic. They are quiet. They appear in passing, a detail of summer, a taste of autumn, a jar in winter. Culture adds layers. Biology provides the foundation. And as we step back from tradition and symbolism, we return once more to the quiet consistency of the fruit itself. Small, predictable, ripening, nourishing. A simple form repeating across generations. Now we begin to soften the edges of this journey, gathering together what we have traced from forest floor to laboratory to kitchen shelf. At the beginning, we noticed something small, a berry resting in the palm of a hand. From there, the path widened. We followed early humans scanning the ground for color. We looked at flowers turning into fruit and how a true berry forms from a single ovary. We traced pigments like anthocyanins deepening from green to red to blue. We saw how wax blooms protect water-filled flesh. We watched the exchange between plant and animal, sweetness traded for seed dispersal. We moved into human hands, drying fruit in the sun, mixing berries with fat and meat, simmering them slowly into preserves, carrying vitamin C through winter months when fresh leaves were gone. We paused with fiber, the quiet structure inside softness. We followed it into the microbiome, where fermentation happens without sensation. We looked at antioxidants and polyphenols, and how laboratory tests once ranked berries high in ORAC scores. We stayed measured with the science, promising signals, ongoing research, careful conclusions. We zoomed in on the brain, on processing speed and memory, where some trials suggest blueberries may support certain cognitive measures. Not miracles, not final answers, just steady investigation. We stepped back again into tradition. Elderberry syrups, cranberry studies, bilberry stories, lingonberries in Nordic kitchens, cloudberries in short northern summers, berries in wreaths and harvest rituals, berries as symbols of abundance and endurance. And through it all, one pattern remained consistent. Berries follow predictable systems. They flower, they ripen, they attract, they disperse, they nourish, they soften. They return the following year. There is something calming about that rhythm. Small chemistry unfolding on a branch, color appearing gradually, sugar rising slowly, seeds forming quietly. Nothing rushed, nothing loud, just cycles repeating at a scale that fits easily in your hand. If you imagine a bowl of berries on a table, you can almost see the layers we explored resting together: botany, ecology, preservation, digestion, culture. Each fruit holding threads that stretch outward into time. And now, as the bowl slowly empties, one berry at a time, the topic can settle into the background. Small fruits, slow chemistry, quiet consistency. Let the details soften, let the colors fade gently, and let the rhythm of ripening and rest carry you towards stillness.